All right, guys. Just give me a minute here. Just wait a minute while I'm setting some stuff up here. A little demonstration we're going to do. Well, I hope everyone's doing okay. A little coolish and windy out there, even though there's supposed to be a high of 19 today. All right. So today we are going to continue on with our adventures about the neuron. See a little chat here. Oh, perfect. Glad to see you here, Ashley. So we're going to continue on with our adventures with the neuron. So we're going to talk a little bit more about that. And then we're going to look at sort of how a neuron responds and how we need a minimum level of stimulus in order for a neuron to respond. And then we're going to take a look at probably starting synops. I don't think we'll get to the end of synops. So I will have a worksheet for you. I haven't given that to you yet. I'm going to just adapt it depending on how far we get for today's class. So we'll see where we're at before I give you that. So your previous worksheet was due today at noon. So please get that to me as soon as possible. And then this next worksheet will be due Friday. And then Friday we'll talk about synops and finish the end of, hopefully the end of the neurons off on Friday. That's the goal. We may not. So I'm thinking, depending on Friday, we'll probably have a quiz on neurons Tuesday or Wednesday. Okay. It won't be on Monday because I don't think we'll finish everything on Friday. Because neurons, there's quite a bit of material for us to cover. Okay. So that being said, let's just move on and talk about neurons. So let's just do a little bit of review before we start. So let's think about, I think I've got it set for everyone. Can just come in here. Okay. Oops. All right. So let's think about how we have our neurons. So remember, we are sort of creating a neuron. So if I'm starting with a neuron, what are my fingers representing? So let's do a little bit of chat. What's my fingers? What are my fingers representing? Okay, so my fingers are the dendrites. Okay, and what's important about the dendrites? What do the dendrites do? Okay, what's important about these guys here? What's their job? Yeah, their job is to receive the information, good, and to send the information to this part, the palm of my hand, okay, which is called so what part of the neuron does the palm of my hand represent? The cell body, good. So if I was going to recognize the cell body, if I was going to take a look at a picture, an image of a neuron, how would I recognize the cell body? What does it have in it? What does the cell body have where I would recognize it? Yeah, it's got a nucleus, exactly right. So the cell body's got the nucleus. Okay, so the message is starting from my dendrites, it's going to travel through the cell body, and then it's going to make its way through this next tube, which is called what do we call that next tube? The axon. Awesome. Okay. Now the axon is going to have these little branches at the end, and every branch is going to end with a little ball. What do we call that little ball? Each branch of the axon is going to extend out to a little ball. What do we call that little ball at the end of each branch at the end of the axon? Anybody remember? Good. So axon terminals, also known as synaptic knobs. Right, synaptic knobs are axon terminals. And remember, it's from those terminals that the next message is going to be sent to the next neuron with its dendrites, or to an effector, a muscle cell or a gland. Okay, so we want to remember that. Okay, let's think about the myelin sheath. What type of cell makes up the myelin sheath? What type of cell makes up the myelin sheath? Schwann cells. Schwann cells, good. And what is the number one most important thing about the Schwann or the myelin sheath? What does it do for that neuron? Why is the myelin sheath important to the neuron? What does it do? What is its main function? Excellent. Speeds up neural transmission. Yes. It helps the message transfer quickly. Exactly right. Okay. And it helps the message travel quickly because the messenger can go from the gaps between the myelin until it gets to the message, right? What do we call those little gaps between the myelin? Or myelin, I guess I should say, myelin. We call those gaps between the myelin. Good. Nodes of Ranvier. Excellent. Okay, so parts of the neuron, right? We have to know all of those things. And then we talked about the reflex arc, okay? What is the first point of the reflex arc? First thing that we need. Excellent. Sensory receptor. The sensory receptor. Next point. Sensory neuron. So this sensory receptor receives the message. Sensory neuron gets it from the sensory receptor. Sensory neuron sends its message to the third point of the reflex arc, the interneuron. Where is the interneuron that receives that message for the reflex arc? Where is it? Excellent. It's in the spinal cord. Awesome. Okay, so now we go from the interneuron and it sends its message to the motor neuron, and then the motor neuron is going to send its message to the fifth point, the effector. Perfect. Okay, so we have those 5 points of the reflex arc. We have to know those. Now, let's say that we touch that hot stove. So we're a little 2-year-old, 3-year-old, and we touch the top of the hot stove. Our receptor detects the pain. Do we feel pain immediately? Right, not yet. We don't feel the pain yet, right? The pain, remember there's 2 interneurons in our spinal cord, one that's going to whip through to the motor neuron, so we get that message to the effector and withdraw our hand really quickly, and then the other interneuron's going to our brain. So it's slightly delayed when our brain's like, oh, that hurts, right? Happens a little bit slower, so we want to remember that with the reflex arc. Okay. Today, we're going to just review a little bit of what we did before when we talked about neural transmission, and then we're going to continue on with repolarization. I'm going to talk a little bit about that. So let's open up your PowerPoint here and get started. Okay, let's move backwards here a little bit. So we'll go through a few of the things that we talked about already, and then we'll this is probably just as good a spot to start as any here. Okay, so we'll just review a little bit. So remember, we have the sodium potassium pump. So we want to remember that that pump is always pumping no matter what, okay? So it's always pumping sodium ions to the outside of the neuron and potassium ions to the inside. So always we have the pump. Remember, pumps go against the concentration gradient and therefore require ATP. Okay, and we took an image and looked at that. We want to remember there's diffusion channels. So when the channel opens, the diffusion channel for sodium opens. That means the sodium ions can go through the neuron membrane. Remember, the sodium is being pumped to the outside of the cell, so it's going to diffuse whoosh, to the inside. Okay, so we want to remember the direction the ions move when they're pumped and the direction the ions move when they're diffusing. Okay, so that's really important for you to remember. Potassium ions, because they're being pumped into the cell, they're going to diffuse out, okay? So resting membrane, we call it polarized. So usually we use that term polarized to talk about when there's a difference of charges, right? Positive on one side and negative on the other. But in the case of talking about the neuron, polarized is referring to being positive on the outside and negative on the inside. So that's what polarized means. And to get this state of being polarized, which is our resting membrane state, that means our sodium diffusion channels are closed. So when the neuron isn't being stimulated at all, the sodium diffusion channels are closed, okay? Some potassium channels are open, not all of them, but some of them. So potassium's busy being pumped into the cell, and then some of the potassium's diffusing out, okay? So because sodium's being pumped out, can't get in, and potassium's diffusing out, some of it's diffusing out, we get more positive sitting on the outside than we have on the inside. Not forgetting that the pump is always, always pumping, okay? So that's our resting membrane potential. And then we talked about when a membrane is depolarized. So that means it's stimulated, which means the charges are flipped. So now it is positive on the inside and negative on the outside. This happens when the neuron is stimulated, and we'll talk more about that with the synapse. But when the neuron is stimulated, that means that sodium channels are open. So sodium's pumped out because the pump is always pumping, but sodium diffuses. Whoosh, we have this wave of sodium diffusing inside the cell, okay? So it rapidly diffuses into the neuron. Potassium channels close, so potassium, while it's being pumped in, it can't get out. So sodium's quickly diffusing in, and potassium is being pumped in, and we end up with a net positive on the inside, okay? If there is enough stimulus, then that'll cause enough sodium ions to come on the inside, and that'll lead to what's referred to as an action potential, right? So an action potential is when we end up with a wave of depolarization happening through the neuron, okay? So, and again, this is much easier with a class than it is with just me, but if I have a wave, if the stimulus is coming from over here, so we want to imagine, okay, so we're just going to look at it this way. We want to imagine that me with my hands at my side, just move this back, like this. Me with my hands at my side, this is me resting, okay? This is me depolarized. This is me resting. So I'm in my resting state. So now that wave's coming towards me, sodium ions are opening, sodium ions open, whoosh, right? Become depolarized. So we have this wave of depolarization that's occurring, and that's that action potential as it's traveling through the neuron. So one area where the sodium ions are rushing in stimulates the next area for sodium ions to rush in, and then the next area, and so on and so on, okay? All right, so now we get to this last state of the neuron, and that's called repolarization, okay? So we say that this membrane is repolarized, but really when we think about repolarized, this is a continuum. So let me explain using my analogy of the wave, okay? Here's me polarized, my hands are at my side, okay? This is me depolarized, okay? So now that area of the neuron is stimulated, but here's what the problem is. If this is stimulated, right, lifting my arms is stimulated, I can't be stimulated again. I can't go through the process of lifting my arms to be stimulated again if they're already stimulated. Repolarization is the recovery. So that's us going from this state of being depolarized back to being polarized. That's what repolarization is, okay? We're here and we're going back to here, because the only way that a neuron can be stimulated again is if it recovers, okay? Because once your hands are up, they can't go up again because they're already up. They have to recover and then they can go up again, okay? So the resting membrane potential of the neuron is restored. So that's the point of going from here, back to polarized. So depolarization of an area of the neuron stimulates. So once an area is depolarized, if enough sodium channels open up, then the sodium channels in that area close, and the sodium channels in the next area open. Okay, so sodium can't enter the neuron anymore. All the potassium channels in that area open up. So what we end up with, so we start wave of depolarization, right? Sodium ions are rushing in, and then we have a wave of repolarization where potassium ions are rushing out. Okay. So potassium rapidly diffuses to the outside of the neuron. Okay. So what we want to think about this as is a wave of depolarization rather, followed by a wave of repolarization. Because when we see the wave done, if we're thinking about a stadium, right? The wave happens, everyone's arm goes up, but right after your arms go up, your arms go down, right? So wave of depolarization, wave of repolarization. So that's when we're thinking about the wave happening. A lot easier to demonstrate when I have a whole class of students, but nonetheless, we've seen this enough that you guys can get my point here, okay? So what's happening, because the sodium channels close with repolarization, then what that means is the sodium can't diffuse in. So we've got all of this positive charge that's sitting inside the neuron, right? And the sodium can't diffuse out because it doesn't diffuse out, can't come in. Potassium channels are open, so potassium is diffusing out, pumped in, diffusing out. But we want to maintain that positivity on the outside. Eventually, as that pump is pumping, it's going to pump the remaining sodium ions back to the outside. And that's where we get that positive state on the outside again. Okay, so we want to remember repolarization is a continuum going from depolarized back to polarized. Okay, so at the end of the day, we get a net positive on the outside of the neuron and a net negative on the inside. That's where we end up. Okay, once we're back to a state of being polarized, then that neuron is ready to get a signal again. It's ready to be stimulated again. Okay, so let me draw a picture of that. So I've drawn a picture of the other two states. So let's draw a picture of this one here. So I'm just going to go to my whiteboard here. Excuse me. All right, so when I'm thinking about this, what I have is, there's my neuron, okay? So I've got the inside, and I'm just thinking about the axon because it's easier to draw, right? And the outside of my neuron, okay? We don't want to forget that there's a pump that's always pumping. So no matter what, whether it's polarized, depolarized, repolarized, or this other state we're going to talk about a little bit later, hyperpolarized, doesn't matter. This pump is always pumping, and it's always pumping sodium to the outside. And it's always pumping potassium to the inside. Okay, so that's always happening. Okay, no matter what. Now, as soon as that wave, right, we have this wave of depolarization, sodium's rushing in, but as soon as that rushes in, that gives a signal for that section of the axon to stop allowing the sodium to rush in. So then we think about, we've got these sodium ion channels here, right? Our sodium ion channels are closed. Okay, so our sodium ion channels, oh boy, I can barely read any of that. Anyway, they're closed. We can see that from my image. So sodium channels are closed. So we want to remember that we were in a state here where we had a whole lot of sodium ions, whoops, a whole lot of sodium ions here, right? Because remember, they'd all diffused inside the neuron. So we've got tons and tons of sodium here. And we want to remember that our potassium channels were closed. So we had been busy pumping all of this potassium inside, and the potassium couldn't get out, right? But now that we're in this state of being repolarized, the sodium channels have closed, the potassium channels have opened. And not just some, right? So when we're in a polarized state, only some of them are open. But when we're in a repolarized state, they're all open. So we have this massive amount of potassium ions that are being diffused to the outside of the cell, okay? So lots of potassium are moving, they're diffusing to the outside. Sodium's being pumped to the outside, and after a period of time, we're going to get, again, our net positive on the outside and a net negative on the inside, okay? So remember, at the end of depolarization, our membrane potential was around positive 40 millivolts. With repolarization, we're going to go back down to our negative 70 millivolts, right? So we want to remember repolarization is a continuum, getting us from this point, right, depolarized, back to polarized. Okay. All right, any questions about that? Okay, so I'm going to bring up the PowerPoint again. We'll go back to that. Actually, I'm going to just bring up something else here first. Just to show you what I haven't posted this yet, but I will, just so you have it to look at. This is something I did a couple of years ago, back when I had a smartboard. But it does give you just some images, just like what I drew out for you, except they're a little clearer here. This is what I have with our state of our membrane. So resting is polarized, right? We see the sodium channels are closed, potassium channels are open. Our pump is pumping. And then here we see a depolarized membrane. And then here we see our repolarized membrane, okay? So this is just there, just as another reference point. I've already drawn these pictures for you. I don't have potassium and sodium shown in my sodium-potassium channel in this first one, but we want to remember sodium's being pumped out and potassium's being pumped in, okay? So I will share this with you if it's helpful to you at all. Okay, and then I'm going to go back to our slideshow here. Like so. All right. Okay, so again, the thing to remember is a neural membrane we can't have a second action potential until repolarization has happened. So that's a really important piece to this. Okay, so a wave of depolarization is followed by a wave of repolarization. So let's just take a look at this little image here, okay? And I'm just gonna draw a little picture here, like so. Just get this up here. So let's say that our neural transmission is happening this direction, okay? So it's going from left to right. So if we're going from left to right, and we're just seeing what's happening right here in this section of the neuron, so that's what I'm focused on, that very first section. What ized word, so when I say ized word, I'm talking about polarized, depolarized, or repolarized. What ized word am I going to use to discover or to describe this region? Okay, so just use your chat to let me know what ized word would I use to describe this circular region right here. So that section. Right, so that section, because nothing's happened to it, right? So this wave, this action hasn't reached it yet. So this is still polarized, okay? So that's polarized. You know what, I'm just gonna write a P there because I think that'll be easier for me to draw. This is polarized, okay, that's a P. What about this section? What would I say about that section? Okay. This one right here. That one we would say is depolarized, okay? And the reason why I know it's depolarized is because I see that rush of sodium coming into the cell, okay? So this section is my depolarized section, so that's my D. Because what we want to remember, what is going on here? Hold on 1 second here. Oops. Okay, I'm just going to, sorry, I'm having some technical difficulties here. Just give me a sec. Just want to go like this. Stop share for 1 second. Okay. So what we want to remember is we have this wave of depolarization that's going on, right? So with this wave of depolarization, it's slowly making its way from one end of the neuron to the next. So the neuron it hasn't hit yet, that's polarized. The neuron where that region is hit, that's depolarized, okay? All right, so now, let's see if this will work for me. Okay, perfect. So now I go over here, and we're going to look at the second image here. Okay, so now again, what I want to do is I want to talk about those eyes words in that second image. So what, and remember, we're going this way, our neural transmission is going this way. So what would this section be? What would I call this section here that I put the X on? What eyes word? Yeah, polarized, because that wave hasn't reached it yet. Perfect. Okay, what about this middle section? What would I call that? Right, so that's depolarized. That's the part that's this part, right? So here, the wave hasn't got to them yet, right? The wave's reached this part. And what about this last part here? What would I call that? Yeah, that's repolarized. That's the part that's recovered, right? So the wave's coming, okay? So these guys haven't got it yet. Oh, wave hits that section, and then we put our arms down, we recover. That's repolarized, okay? All right, so we want to make sure that we can identify those different regions, whether it's polarized, which means the action potential hasn't hit it yet, whether it's depolarized, which means this, right? Sodium ions have rushed in, or repolarized, which means it's in the point of recovery. It's starting to recover, okay? All right, so there we are. This is just another image of saltatory conduction. So that's just the jumping of the message from node of Ranvier to node of Ranvier to node of Ranvier. So that's saltatory conduction. And that's what we see happening here. So we see the messages jumping from this region, which would be the node of Ranvier. So that's where we have our diffusion channels. And then diffusion channels are here, and then diffusion channels are here. So we don't really have any movement that's happening here where the myelin is, okay? All right, so I have a couple videos that we're gonna watch. Hopefully this will help with your understanding of how this process all works. So let's just go to the videos here. And that. Share the sound. Okay, so let's start with this one.
An action potential, depicted as a red band, is propagated in one direction along the axon. During an action potential, the inside of the cell membrane becomes positive with respect to the outside. An action potential generates local currents that tend to depolarize the membrane immediately adjacent to the action potential. When depolarization caused by the local currents reaches threshold, a new action potential is produced adjacent to the original one. Action potential propagation occurs in one direction because the recently depolarized area of the membrane is in absolute refractory period and cannot generate an action potential.
So what I like about this is we can see that that red area, whoops, cancel, back.
An action potential.
This little red area that we see down here, this is our wave of depolarization as it's traveling through the length of the neuron, right? So wave of depolarization, and it's quickly followed by that wave of repolarization. So that's what's
positive with respect to the outside.
Kind of neat to see when we're looking at this, right? So we see this wave, this region where it's going to be more positive on the inside. That's telling us it's depolarized. and it's followed by this region where it's more positive on the outside. So remember he said absolute refractory period, nothing can happen there because it's in the point of recovery. It's in the point of going from depolarized back to being polarized again. Okay. All right, so let's look at the next video here. It goes into a little bit more detail with the channels. So it's this one.
Pulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges.
Okay, so that's because the sodium ion channels are closed. Some of the potassium channels are open, so potassium can move back out through the diffusion channels.
The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open.
So the hint here is the change has to be enough, and we're going to talk about that. It's a concept called threshold level. We'll get into that in a minute.
This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to...
Okay, so that's the wave of depolarization. So when one region has that depolarization, it spreads to the next. Again, thinking about that as a wave when you're going to see a gain.
Restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive.
Okay, so suddenly sodium channels
This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
So that wave of depolarization is followed by a wave of repolarization that we can see in this image here. Okay. All right, the last video we're going to watch in this little section is on saltatory conduction. So I'm just going to cancel that. Like so. And this one.
Conduction. Conduction through myelinated nerve fiber. Due to the myelination of neurons, action potentials may only occur at the nodes of Ranvier. Myelin is made up of insulating cells, which means depolarization cannot occur in myelinated regions. Between these cells, however, there are gaps known as nodes of Ranvier which are unmyelinated. As depolarization cannot occur at the cells making up the myelin sheath, the wave of depolarization can only occur at the nodes of Ranvier. Thus, action potentials appear to jump from node to node when traveling down an axon, and this phenomenon is known as saltatory conduction, and it serves as a means of increasing the rate of propagation of an action potential. Not only does saltatory conduction increase the speed of impulse transmission by causing the depolarization process to jump from one node to the next, it also conserves energy for the axon as depolarization only occurs at the nodes and not along the whole length of the nerve fiber as in unmyelinated fibers. This causes up to 100 times less movement of ions that would otherwise be necessary, therefore conserving the energy required to re-establish the sodium and potassium concentration differences across the membranes following a series of action potentials being propagated along the fiber.
There we go. Okay, so we will end that.
You know what it feels like. You've been studying hard in med school.
There we are. Okay, let's go back to the PowerPoint. There we go. And this. Okay, so now questions. So let's go like this. So question number 1. The sodium-potassium pump works to pump sodium ions where? Into or out of the cell? The pump. Okay, we got a lot of outs here. Yes, out of. Good job. Okay. An unstimulated neuron, so a resting neuron, has more positive or negative charges on the outside of the cell. Which one? Good. When it's resting, it's more positive. Okay. Next one. During a resting membrane potential, the neuron is permeable, so that means the ion channels are open to potassium or sodium. Which one? Right. When it's resting, it's open to potassium. Okay. Next. During an action potential, so that's where we have our wave here. The membrane of the neuron is, so what I's word are we using for that section? Polarized, depolarized, or repolarized? During an action potential. Depolarized, that's right, depolarized. Okay. During an action potential, right, so when we're in this state right here, the neuron becomes permeable to blank, and these ions move blank. So permeable to either sodium or potassium, and then they move into or out of. Good. So more sodium ions are going to move in. They're going to rush in when those diffusion channels open. Excellent. Okay. During an action potential, the inside of the neuron becomes more, what charge? Excellent. Becomes more positive. During repolarization, so now this is our recovery, the neuron is permeable to what ions and impermeable to what ions? So it can pass through. So when I say permeable, I'm saying the ion channels are open. When I say impermeable, the ion channels are closed to that ion. So when it's repolarized, it's open to potassium, good, and closed to sodium, good. All right. Okay, we have to move on here to threshold level. So when we talk about whether enough sodium ions get in, that's what we're talking about threshold level, okay? The minimum level of stimulus required to produce a response. That's our threshold level, okay? So what this means is there might be some stimulation that causes some sodium channels to open, but it's not enough to cause this wave of depolarization. So basically, we might have some stimulation right at the end of one dendrite, but it's just not enough to cause the wave. So it just fizzles out. There is no wave, okay? It hasn't met that minimum intensity level in order to cause a wave. And this part here says the threshold level varies for every neuron. Okay, so let's just take a look over here. So I'm going to just open up my screen so we can see what I'm doing. I'm going to stop the share for 1 second and show you my dominoes. Okay, so I'm going to move this so you can take a look at what my dominoes look like on my table. Boy, that's hard to see. let's make this a little easier. Here. Okay, so what I have is dominoes. Okay, so there's my little row of dominoes. And we want to imagine these dominoes are kind of like an axon. Okay, so these dominoes are like my axon. If I stimulate the very first part of that axon, right, I can make that first domino wiggle. Okay, like so, but nothing else happens. Okay, so I can make it wiggle, but it's not enough to cause a wave of depolarization through the length of the neuron. So nothing's going to get passed through. So it's like we have a little bit of sodium ion channels open, but nothing else happens. But if I have enough, if I meet threshold, we get a wave of depolarization that passes through it. Okay, so that's what that means, is we need to meet that minimum, or we don't have the wave of depolarization. And that minimum is our threshold level. That's what that's called. Okay, so we don't reach the minimum. So where have we experienced that? We can think about things that are out there that we know exist, but we don't perceive them. So for example, any kind of light that's outside visible light, we can't perceive. So we can't perceive ultraviolet and we can't perceive infrared, which are the other two sides of our visual light. Okay, we can't see that. So it exists, and it may cause a little bit of sodium ions to go in, but not a wave. We don't perceive it. Okay, or if we have a frequency that's really high, right, like a dog whistle, a dog can perceive it, and it exists. So again, it may cause a bit of stimulation just on the very initial part, but not enough sodium ions go in that it doesn't cause a wave of depolarization. Threshold has to be met. Okay, so we'll just quickly go back to the PowerPoint here. We want to understand that that threshold level varies for every neuron, right? So some neurons have a higher threshold level and some neurons have a lower threshold level. So you may have heard that term pain threshold, right? Having a high pain threshold or a low pain threshold. Basically what that means is if you have what's termed a high threshold level for pain, then that means you need a lot of pain stimulus in order to feel the pain. If you have a low threshold level for pain, you don't need very much stimulus in order to feel the pain. Okay, so it takes a lot. If you have a high pain threshold, it takes a lot of intensity in order for a wave of depolarization to occur. A low pain threshold, very little intensity. Okay, now that's different. That's a different concept than pain tolerance. Pain tolerance is how good you are at tolerating pain. So different people have different coping mechanisms to tolerate pain. That's different than perceiving pain. Okay, all right. So that just sort of relates that whole idea of threshold to what we were talking about. I think we're going to do the next two slides and then we'll stop. Okay, I do want to talk about this graph a little bit. Okay, so what we see here is this is a very common graph that we use when we're talking about membrane potentials. Okay, so that's this graph right here that we're looking at in the middle. And there's different parts to that graph. So if we look at part one, that's this section right here. Okay, so there's part one of our graph. That's the resting potential. And we see over here what's happening with part one. We see it's more positive on the outside and more negative on the inside. So if I look at part one, let's just go to the chat here. What ion channels are open in part one of this graph? What ion channels are open? Okay, this is our resting membrane. It's resting. The only channels that are open are the potassium channels. Okay, so the potassium channels are open here. If I go to part two, I see that some sodium channels have opened, right? So some sodium channels have opened, right? So we have that little bit of stimulus that's happening. If I go to part 3, so that's what I'm seeing up here. In part 3, enough sodium channels have opened that are causing a wave of sodium channels to go in, and that bumps us up here, right? So I said it's about plus 40 when we reach that top of the action potential, okay? And then as that wave of polarization happens, it's followed by a wave of depolarization occurs. It's followed by a wave of repolarization, right? And this is where our sodium channels are closing and all the potassium channels are opening, okay? So that's what I'm seeing over here. Sodium channels close and all the potassium channels open. But here's something different that I haven't talked about yet. I see a little dip. And what that dip means is we've kind of overshot that positive, and it's become too positive on the outside. So let me just quickly explain that, okay? So what I have here is, here's our neuron, right? Here's our pump, and it's pumping sodium ions to the outside and potassium ions to the inside, right? And when we're recovering, right, all of our potassium channels are open. So potassium is just rapidly diffusing out. But because they're all open, we have this huge amount of potassium that diffuses out, which means we get not just polarized, but more positives on the outside than we normally have. Well, when we have more than something, right, we call it hyper. So this state where we have too much positive on the outside is referred to as hyperpolarized. So that little section of the graph that we were just looking at is hyperpolarized. Too much positive sitting on the outside. Okay, so we'll just go back to the PowerPoint quickly here. Okay, so it overshoots it, so we have too much positive, but remember that pump is going to be pumping that potassium back in, and eventually we'll get back to what we're seeing right here. Whoops, sorry guys. What we're seeing right here, which is our resting membrane potential. Okay, all right, so we're just going to quickly look at that next image, and then we'll be done with this here for today. All right, so just open that up so I can see what you guys are saying here. So we're going to indicate, so what I want you guys to do here, indicate the state of the membrane at each of the numbered regions. So when I say state, I'm talking about the ized words. So our first one here is, get my spotlight going, is number 1. So that's this line right here. So what ized word would I use for number 1? Yeah, so number 1, that membrane is in the state of being, so we're looking at right here, the state of resting membrane or polarized. Okay, what ized word am I going to use for number 2? Okay, what ized word? It's depolarized, right? We're going like this. Okay, what ized word am I going to use for number 3? Excellent. So number 3 is repolarized, but now, oop, we have a dip. What ized word do I use for that dip? Okay, we've overshot it. Hyperpolarized. Perfect. That's hyperpolarized. Okay, so polarized, depolarized, repolarized, and hyperpolarized. All right, guys. So we're going to call it a day with that. Within the next hour or two, I will share the worksheet that's due on Friday's class. Okay, or on noon on Friday, so before Friday's class. All right. Any questions before we sign off? All right, so we'll see you guys on Friday. So have yourselves a wonderful next couple of days. Just a quick note, for those of you that are in grade 11, make sure that you're dealing with your registration. You should have been doing that with your Block 2 teacher. If you don't have a Block 2 teacher, get in touch with Mr. Hallbrow right away. So if you're in grade 11, Or if you're in grade 12 and planning on taking courses next year, if you're in grade 12, talk to Mrs. Hamilton right away about registration. And if you're in grade 11, talk to Mr. Hallbrow if you have not heard from your Block 2 teacher, okay? So make sure you get that done because you want to get into the courses that you want to get into, right? And have a great 2 days, and we will see you guys on Friday. All right, bye-bye. Threshold levels, will there be any symptoms that someone can experience from an extra low or high threshold level? There is a condition, I'm not sure if it's threshold levels or the pain receptors aren't working. There is a condition where you don't feel pain, and that's bad. That's incredibly bad because you don't respond to pain. Pain tells us to stop, and then you don't respond to it stopping. I haven't heard about having really high, or sorry, that was high threshold, having low threshold. That would be something that I'd have to look at, because there's a difference between the neurons having the threshold or the neurons or the receptors not functioning, and I'm not sure which. The symptoms would probably be the same. With a low threshold level, would you get spasms? Maybe, because you'd have too much, right? You get, so if your threshold level was too low, you might get overstimulation happening. So yeah, that could be a plausible outcome to that, absolutely. Yep. Anything else? Yeah, that's as good as I've got, Eric. All right. Okay. Have a great couple of days. We'll talk to you later.